It's really wonderful to present to you a major scientist who's been a leader in genomics, whose work has been the basis of understanding cancer and has led us to understanding treatments in both cancer and infectious disease. It's my great honor to introduce you to Eric Lander.
Now, how's that for a flash mob? That's great. Let's hear it for them. Woo! Thank you, Kitty. It's absolutely great to be back in the music tent here at Aspen. The Ideas Festival is just a wonderful, wonderful event. Now, this is an afternoon that's filled with music, some artists, and a lot of political leaders, and a scientist. So what am I doing here as a scientist? Well, Walter Isaacson and David Bradley and I had been talking about a bunch of things and about the remarkable state that biomedical research is at. And they both said, come give dispatches from the frontiers of science. And so that's what I want to do. I just want to take a little time and give some dispatches about what's going on, because over the last 4 or 5 years, there has been just this remarkable revolution that is beginning to gain steam, and I bet it's not on your radar. It's really not on most people's radar. It has the potential to have a huge impact on biomedicine, to accelerate, I think, progress by 25 years. But it's only a possibility. It could do that. The sort of choices that we have to make are pretty important as to whether we're actually going to get the benefit from that. So I want to tell you about the science, and then I want to talk about what we have to do to make sure that we can actually get the promise of this. So that's the plan. It might properly called, what I'm going to talk about, the real Human Genome Project. But to tell you what the real Human Genome Project is, I have to first go back and tell you about the old Human Genome Project. So let me take a moment and tell you about the old Human Genome Project. Oh, somewhere around the mid-1980s, scientists began talking amongst themselves, maybe we might be able to read out all the information contained in the human chromosomes. That's about 3 billion letters of DNA information. 3 billion letters. Let me see if I can put that in some context. I was working on a little bit of DNA sequencing then, and you could get about 200 letters done in a good week. If things didn't work well, you got about 0, but if it worked well, you might get 200. And if you thought about the rate of production of DNA information across the entire scientific community, you could calculate that we could probably do the entire human genome in somewhere between 2 and 3 centuries. Now, this was not an attractive sort of proposition for young people going into a field. It's not that attractive for granting agencies, things like that. I'm in favor of a long-term perspective, don't get me wrong, but this was not a very attractive problem. When you thought about the costs, you know, the best guess of what the cost of doing this was about $10 a letter times 3 billion letters, might be $30 billion, seemed pricey. But this idea captured the imagination of a lot of young people, myself included. I was a young person then. And I got involved in this, started one of the early genome centers that was at MIT. And by 1990, the human genome community had been debating this, and by 1990, we thought we probably could just manage to do this thing in 15 years if we created all sorts of new technology and worked together in all sorts of very unprecedented ways, but it might just be possible. So we started this center. It ended up being a flagship center of the project, did about 1/3 of the project. But what I was most proud of was this was a project done by groups around the world. Groups in 6 different countries, the United States and the United Kingdom, in France and Germany, in Japan and in China. Everybody working under a common goal to make all this information freely and immediately available on the web every night as soon as it was generated. And the groups worked together to do this. And quite remarkably, it worked. In June of 2000, a draft sequence of the human genome was announced at the White House.
It was still drafty. It was only about 90% of the genome. It had a lot of holes, and it had errors here and there, little moth-eaten in places. But it was 90% of the genome, and you could begin to see that whole landscape. And this international consortium stayed together for the next 3 years and worked all the way to get a finished sequence of the human genome. Really high quality, very few remaining gaps. It got completed April 25th, 2003, and the date was chosen for a specific purpose. It was the 50th anniversary to the day of the publication of Watson and Crick's paper about the double helical structure of DNA. And so we all stayed up late to make sure we could make that deadline. Well, look, it was in fairness, a tremendous scientific accomplishment. Everyone's been very proud of it. For the first time, we had the full sequence of the genome before us, and we could begin to analyze things, and it would be a foundation. Of course, it's also fair to say there was a lot of hype around this. The New York Times trumpeted in full-page headlines about the human genome, and, well, people were promising that we were going to cure everything really soon. This was nuts, just certifiably nuts. We had the sequence of the genome, but we had the sequence of one person. We didn't really know how to read it. We had all the letters in front of us, but it was a language we didn't actually speak. And, well, even if we understood how to read it, and even if we knew how to convert it to find the causes of disease, and even if we knew how to make medicines, it would still take a decade to get all those medicines through the clinical trials. And so the idea that the human genome was going to lead instantly to cures was, I think, quite naive. And right on time, the New York Times published an article some years later, again on the front page, announcing all diseases not yet cured despite Human Genome Project. You always love this. But now, in fairness, there is a law of technology. And the law of technology is that we grossly overestimate the short-term impact of technological advances, but we underestimate the long-term impact of technological advances. And that's really what we've begun to see, and that's what I want to talk to you about, is the decade after the Human Genome Project and where it leaves us today and the choices we have going forward. So, one human genome. If we want to learn about disease, we're going to have to compare thousands of people with Alzheimer's or diabetes or different diseases. We're going to have to look at many different kinds of cancers and ask which genetic changes underlie which disease 1 and disease 2. And the thought of doing this at $3 billion a pop times tens of thousands of people, you do the arithmetic, that's tens of trillions of dollars, several-fold the GDP, hard to justify. But over the last decade, something remarkable has happened. The scientific community's come together and begun to figure out how to do this much more efficiently. First, it was ways to skim the genome and get much of the information out pretty cheaply. And then, just in the past 3-4 years or so, the ability to read out the entire genome much, much more cheaply. And when I say much more cheaply, what I mean is this. $3 billion now costs $3,000. It's a 1 million-fold improvement over the course of roughly a decade. I don't know how many million-fold improvements we've seen. Think about if housing prices fell by a factor of 1 million over the course of a decade, not due to market crashes, because somehow we could build houses stunningly more efficiently. Or if transcontinental travel was 1 million times cheaper because of technological advances. Well, that's what we're living through right now, is really in the past several years, it's now 1 million times cheaper to sequence DNA than when we did it in the Human Genome Project. And what that means is amazing things are starting to happen. Let me tell you what I mean by amazing things. Schizophrenia. Schizophrenia is a devastating disease. It affects about 1% of the population, and it onsets at the worst possible time, in early adulthood, with devastating consequences. What we knew about the genetic factors that contribute to schizophrenia just 5 years ago was nothing. 0 genes. Groups got together. I'm proud this group happens to have been a group at the Broad Institute, which I direct. And they began looking at large collections of patients. They looked at 10,000 people, 5,000 with schizophrenia, 5,000 without, and began to do this genome skimming, the light version of it first. And they did this, and they got to the end of it, and they found nothing. This worried them a great deal. In fact, the National Institute of Mental Health said we shouldn't be doing any more of this. You know, this doesn't look like it's working. But when they looked at the data, they said, yeah, nothing's significant technically, but when you look at the data, there are a lot of things that are close to significant. Let's press on. And thanks to philanthropy, they pressed on. And they looked at 20,000 people, and they found 5 genes. And then they took it to 40,000 and 80,000. And now, as of last month, there are 90 genes that have been identified as playing roles in schizophrenia, and they start to make sense. 4 of them are all 4 of the subunits of a particular thing called a calcium channel. And you don't need to be a biologist to say that means something. That says that calcium channel must play some important role. And a bunch more of these genes are involved in something called postsynaptic densities, things that are at synapses of nerves. And if you look at the picture and you squint, you can begin to get the idea that this is going to be a disease about the pruning of neurons at a certain stage of development. And it's going to take some more years to get it. They've only done the skimming part. Actually, the sequencing part has begun for a few thousand, and it's confirming it. But for the first time, we can begin to see causes of schizophrenia. But it's not just schizophrenia. Type 2 diabetes, adult diabetes. We're beginning to see the genetics of that falling out. Early heart attack. Well, we know that high lipids are bad for you, but some people die of early heart attacks and they don't have high lipids. What's going on? Well, again, people have begun to collect large samples and begun to do the skimming, not yet quite the sequencing, and are finding the genes that begin to explain why people die of early heart attacks. Inflammatory bowel disease, a disease about which we knew nothing not so long ago. There's now more than 140 genes arranged into 11 pathways, and it begins to start making some sense. Now, I don't want to overstate that we understand all of it. It's that for the first time, we're beginning to glimpse what the architecture is of these diseases and the pathways in which they fall. Now, in no disease are the implications more important than for cancer. What's been going on in cancer is just remarkable. Cancer is a disease of the genome. What do I mean by that? I mean that cells get mutations in your body. Some mutation happens in one cell in its genome, in its DNA, and that causes the cell to start growing in a certain way. And then it gets another mutation and another mutation, and they begin to pile up these mutations, and that eventually leads to cancer. And you'd think, if I could just read out the information from a cancer and compare it to the normal cells in the rest of the body, I could write down all of the mutations that had happened. Now, some of them are just random noise, but if I start comparing many cancer patients and I ask what's in common, I can begin to see the genes that underlie cancer. Well, in the 1980s, the mid-1980s, people began to use functional biological techniques to find cancer genes, and they found many cancer genes. But what's amazing is over this last decade, using these kinds of tools, all sorts of new cancer genes are being discovered by systematic approaches that didn't require a prior guess as to what these genes do. Genes for lung cancer. It turns out the thing we used to call lung cancer is now a pie chart with many different slices. There's 10% that have mutations in one particular gene, and 10% that have mutations in a different gene, and now it so happens people have developed drugs against those particular mutations for those slices of the pie. Skin cancer. About half of skin cancers have mutations in a particular gene that goes by this funny name called BRAF. And just recently, the FDA approved BRAF inhibitors against that particular mutation. Multiple myeloma. We now know some of the pathways underlying multiple myeloma and brain cancers. In fact, I'll tell you about an experiment that was done 3 weeks ago. 3 weeks ago, someone, a colleague back at the Broad Institute, took all the data from 6,000 different cancers, poured them all into the computer, the DNA information from the tumors and the normal, and asked the computer, how much can you reconstruct of everything we know about cancer based on this information? And the computer was able in a single run to reconstruct almost all the knowledge we have about genes that underlie cancer from that data set and discover many more genes as well. which tells us we're not done yet by far. Now, what's the implications of any of this? Well, it becomes a roadmap for therapy. That skin cancer gene I told you about, 50% of patients have mutation in this particular gene. There's now a drug that attacks the mutant protein, and its effects are nothing short of miraculous. There's a patient, there's an amazing picture, a patient has tumors all over their body. They've received this oral drug, and they melt away. The tumors are totally gone. That's the good part of the story. The realistic part of the story is 12 months later, they're back because the tumor develops resistance to that drug. This is not a simple game. So then you sequence these tumors and you ask, what changed? What trick did they have? And you begin to see that there are some specific tricks they're using. They mutate something else, and it so happens in this case there's a drug against the something else, so you say, let's combine the two. And then they seem to work much better with the pair, at least in preclinical trials. How do we treat AIDS? We treat HIV right now with a triple drug combination. Any one of those drugs, AIDS will develop resistance. Any two of those drugs, AIDS will develop resistance. When you get to 3, the chance of resistance to all 3 is so low it doesn't happen. The way we're going to win cancer is we're going to win it with combination therapies. The only way we're going to do that is having the full picture, the big picture, the complete roadmap of the mutations, and knowing that if we hit here, it's going to try to escape here, so we also better hit at this place, and take it on in a kind of pincer movement. That brings me to my point. Big data, big information. We know the power of big information, big data. When you go to Google Translate, and you type in something into Google and you want it to translate it into French, it's not that Google Translate knows French. It's just that Google Translate has a huge corpus of texts in English and in French that line up against each other, and it says, a little phrase here, let's see, when that phrase appears in an English text, it corresponds to a text over here in French, and it does an amazing job with that because there's enough data. We're now at the point where we can begin to start doing this for biology, where we can have large enough information to be able to look up what are the causes of the particular diseases, how do they mutate to get around particular drugs. So what we really need is not the old Human Genome Project, which was fun in its own way. This old Human Genome Project was about one human, one person's genome. Actually, it was a guy from Buffalo, but we won't discuss that now. What we really need is a real Human Genome Project that involves humanity coming together to do this. It'll involve tens, hundreds of thousands of samples. It'll involve, in the long run, millions of samples to be able to find causes of disease, to be able to make the pathways, to be able to point industry with roadmaps to where drugs need to get made. Is it going to happen? There's no scientific reason we can't do this. But there is a real choice, and that's one of the reasons why when David and Walter asked me to come speak, I did want to talk to you because there are potential obstacles to our doing this, and I just want to name them. There are 4. The first, would we be able to use this information for the benefits of patients? By which I mean, would we have the legal right to be able to sequence someone's whole genome and give them back all that information? Or would we be faced with a thicket of patents, patent protection that would prevent us from doing that large-scale analysis and handing it back to patients? And here I am just smiling because 2 weeks ago, the United States Supreme Court decided in a unanimous decision that human genes cannot be patented. And Aspen is just amazing. You'll have the chance later to thank Justice Kagan as one of the 9 there for that. That's number 1. Number 2, though, the technical ability to combine all of this information, to share information. You might think that should be pretty easy. Think about your electronic health records, if you have electronic health records. They're all incompatible with each other. Even hospitals within the same network can't exchange data often because people have these balkanized legacy systems of electronic health records that don't speak to each other. Can we manage to get the world to come together? And it's got to happen in the next 2 or 3 years so we don't go off in a Tower of Babel of different solutions, but come together around the ability to speak that same language. And here I'm delighted that a number of people, including some of my colleagues at the Broad, brought together 70 organizations around the world in something called a Global Alliance. It was announced just in the beginning of June, where all these organizations have agreed to work together to have common standards and ability to exchange data in one uniform way. And these include the National Cancer Institute, National Institutes of Health in the United States, major hospitals, charities in England, in Canada, in Asia, and it's a remarkable thing. It hasn't happened, but everyone's agreed on paper they're going to do this. Number 3, we're going to need a patient movement, because in the end, every piece of this data belongs to an individual, and it should only be shared with that individual's consent, with their active desire to share. We need a patient movement. I call this thing Count Me In. That's my name for it, because I imagine that when a cancer patient or a mom with a child with some serious genetic disorder at birth comes in, they're given the right to say, I'd like to share the data. Check the Count Me In box. And a friend of a cancer patient says, or some other patient with some other disease, did you check the Count Me In box? Do you want to share? And people are welcome not to. They're welcome not to. But my experience has been most people want to be able to share this, want something good to come of whatever their medical experience is. Of their worst experiences, it makes meaning from it. And I think if we had a kind of patient count me in movement, many people would stand up and say, count me in. And then we have to worry about scientific funding. And I'm sorry to even think about this, but just at the time that we are seeing the payoff for the Human Genome Project and this amazing increase in technology, we're also seeing cutting of biomedical funding. I can't tell you what the effect has been, I can't tell you how serious the effect has been of sequestering 5% of the NIH budget. What it has meant is the scientific community is now getting very cautious about taking on ambitious new projects because they've got to worry about a greatly diminished budget. In fact, in real inflation-adjusted terms, the NIH budget has fallen by 25% over the past decade, while the opportunities have gone up just spectacularly. And so many of us in the scientific community are worried about, is this the right kind of investing we should do? You might say this is woolly-headed scientific thinking investment. The Battelle Institute did a study of what had been the return on investment on the Human Genome Project. And their current calculation in 2 reports they issued was it's returned about $965 billion, just under $1 trillion in economic impact of having this information. So from a purely hard-headed business point of view as a country, we ought to be investing in such things. And I think we will even have the opportunity, thanks again to Aspen, to be able to talk to Eric Cantor about such things. And I got to say, Eric Cantor is a big supporter of biomedical research. I know he believes in the power of all this, and I hope we can figure out how to make sure we support these things. And we'll have to do the same thing. I mean, just think about the signal it sends to our young people right now, whether we are or are not investing to take advantage of these opportunities. Of course, it's not all just the government. Philanthropy has played an enormous role in all of this, because it's only philanthropy that can really take the really bold projects that don't have to go to a peer-reviewed study section for approval and vetting, but philanthropists who can say, I'll bet on it, I'll take a risk. And so that's a truly remarkable thing. I direct the Broad Institute. The Broad Institute exists in large measure because of the Broads, who are also here, Eli and Edie Broad. So anyway, these are my dispatches from the frontiers of science. You're probably not quite reading about all of this yet in the newspapers. And I want to be realistic. I'm not saying we're going to cure everything. I'm a realist. We're not going to cure everything. There will be tremendous progress, but there still will be disease. It is not the case that genes are the answer to everything. Environment, diet play very important roles, although genes can be very important ways to learn about the roles of environment and diet. And it's not going to happen overnight. I told you already about the time frame for understanding, the time frame for developing drugs. I don't do this because I particularly think I'm going to be the beneficiary of it. But our kids could be. If we play our cards right, all of this, all these pieces, these therapeutic roadmaps, and these drugs, and the ways to use them, will be here for our kids. Or if we don't play our cards right, they won't be. That's the choice. That's why I wanted to come talk about it. I thank David and Walter, because I want to be able to count all of you in. Thanks much.